exciting case, and this time I'll introduce Curtis Cline, but I won't tell you where he works. He'll tell you later. Okay, Curtis?
All right, good. Let's see, it's good afternoon on the East Coast, good morning on the West Coast. I'll present a slide, or talk about the slide, and then I'll introduce myself and more formally after. All right, so in this section of Brain from a Mouse, here within the thalamus, the perivascular space is expanded by moderate numbers of mononuclear cells and fewer neutrophils, which extend into sort of obscure the vessel wall architecture. And then what's that? Good. Neuropil directly surrounding the vessel. There's low numbers of migrating microglia, and occasionally in other vessels, there's a few lymphocytes and neutrophils in the adjacent neuropil as well. And then rarely, in some of these smaller vessels, there's pale eosinophilic hyalinized fibrin thrombi, again, partially occlusive. And then in a few of the vessels, there's small amounts of perivascular hemorrhage, which occasionally extends into the adjacent neuropil. And then down here in the hypothalamus, and this feature is most prominent here in the hypothalamus, there are neurons that have a pale clear white halo and are shrunken with hypereosinophilic cytoplasm and pycnotic or occasionally karyorectic nuclei. And again, this feature is most prominent here in the hypothalamus, but also present over here in the ventral striatum. These are interpreted as necrotic or perhaps even apoptotic neurons. And notice there's kind of a conspicuous absence of abundant inflammation in the area, although there are some migrating microglia. And then last but not least, within the meninges, they're infiltrated by mononuclear inflammatory cells, and then directly underlying them in the cortical gray matter, there's migrating microglia, rare neutrophils present as well. And then finally, in this region of the cortical gray matter, there are low numbers of neurons which have pale, almost like a ground glass cytoplasm or microvacuolated cytoplasm that also occasionally have a single clear large vacuole that appears to compress the nucleus. And I interpret this as a degenerative change. And so that's the description of the slide. I called it meningoencephalitis with neuronal necrosis, perivascular cuffing, kind of bordering on the line of vasculitis, and rare fibrin thrombi. So, want to have any guess as to what this is, or want me to go right into what's going on?
Okay, so the differential would be obviously some infectious disease, autoimmune, toxin, several other things. Anyone else have any possibilities? So infectious disease, virus, something else, toxin, chemical toxin, some other thing. Any other ideas out there? Enrico, he's the neuropathologist on board here. Give him a chance. He's chatting only. You got the sound working? No. Okay. Anyway, hey, Tony, you're a diagnostic pathologist. What do you think?
I'm sorry, I didn't have my headset on. I think sepsis would be my first thought, seeing the type of lesions. But I'm open to obviously other possibilities.
Okay.
Did you hear what kind of mouse this was?
I haven't said what kind of mouse it is yet, because I think that would give it away.
Yep. Okay.
Curtis, you know I'm going to ask you about when you say vasculitis, what was it consistent, the caliber and the character of the vessels that you saw there? Were those all veins and not arteries, etc.?
Mostly veins. Yeah, mostly veins. And yeah, it was mostly in the perivascular space, but in some cases there were small amounts of necrotic debris associated with it. And so I went with vasculitis instead of just perivascular cuffing, but this was pretty characteristic of the lesion. In some cases, like I say, there was more debris kind of associated with it. But
Yeah, tough call in a vein, too thin-walled to make that call sometimes, but I agree with you.
yeah, I vacillated on this one a little bit, but I stepped out on a ledge, I guess.
Okay, you can give your PowerPoint if you like.
Okay, so let me share my screen. PowerPoint. All right, we good? See
Now you know.
Okay, so this is what I described as a mouse CNS lesions in a transgenic mouse model of SARS-CoV-2. So my name is Curtis Cline. I'm a pathologist. I work at USAMRIID, the U.S. Army's Medical Research Institute of Infectious Disease. And I'll get in more into the model in just a second. I got to do the DoD disclaimer. The opinions, interpretations, conclusions, and views are those of myself and do not reflect the official policy of the U.S. Army, the DoD, or the U.S. government, the standard disclaimer. So this mouse I just described, this section I just described, is an animal from this study that we published using a transgenic mouse model of SARS-CoV-2. So this mouse expresses the human ACE2 receptor under the control of the keratin 18 promoter. So just briefly, this study, 28 animals divided into two groups, a high dose and a low dose, 10 to the third PFU for the low dose, 10 to the fourth for the high dose. And then the two groups, high dose and low dose, were further divided into male and female. So there were 7 mice total for each of the 4 groups. And okay, all right. So this mouse is from this study. So we saw lesions obviously in the lung, but also in the brain, and then the nasal turbinates and the olfactory bulb. And so this lesion that I'm showing here is pretty characteristic of what we saw in the nasal turbinates. So multifocal areas of flattening and attenuation of the olfactory epithelium. Here, the multi-layered kind of appearance is more normal. And then we saw this multifocally, but it was pretty minimal, or in some cases mild. And then this is the RNA-scope in situ hybridization for SARS-CoV-2. And the black arrows at the bottom of the screen indicate where it was located in the kind of the dorsal nasal meatus of the olfactory epithelium. So this is, again, pretty characteristic. We saw this in the day 3 animals, as far as in the olfactory epithelium, the day 5 through 7 animals. And it was pretty minimal, really. The labeling was pretty minimal. And in some areas, the presence of virus did correspond with the areas of degeneration. But kind of most exciting here is the lesion, or the presence of virus in the olfactory bulb, and kind of present throughout all layers, but most prominent in the like the mitral cell layer. But there's also a little bit in the granular cell layer as well. And then we did the duplex IFA, where we used neuronal nuclear antigen and co-labeled with SARS-CoV-2 just to document that the virus is indeed present in neurons, and all the labels kind of what I just corresponding to the ISH that I showed previously.
So you say exciting, I say scary, but I immediately have a question, which is the molecular basis for this was to humanize ACE2 under control of keratin 14. So how the heck did it get into neurons which don't express keratin 14? And
That is an
I'm sure you're going to talk to me about that later, but I had to ask now.
no, that's an excellent question, and that's we're going to definitely get to that. So we'll definitely get to that. I'll just keep going, and if I don't answer, ask me again at the end, okay?
Fair enough.
Okay, so this is again RNA scope ISH for SARS-CoV-2. This is a similar section to what I described, and abundant virus kind of in the central thalamus, hippocampus, cortex, ventral striatum. Note the kind of strong labeling down here in the ventral striatum, as well as in the hypothalamus. And note that's where I describe those necrotic or apoptotic neurons. And so just the black box here will blow that up a little bit more here. And notice that this labeling here in these neurons, and then also out here in the neuropill, which I believe would correspond with the virus presence in likely in axons. And then one other thing I want to note is this vessel here, and there's kind of conspicuous absence of abundant virus in the vessel, and that's pretty characteristic for what we saw not only in the brain, but also what we saw in the lung. A lot of dense perivascular cuffing in the lung, but no virus really in the vessel walls at all, or in the endothelium. So again, duplex labeling here with GFAP and SARS-CoV-2, the white triangle with labeling of the GFAP there in red. We didn't see any co-labeling, so made the assumption that astrocytes aren't being infected with the virus, and that's kind of in contrast to the image on the right. Again, neuronal nuclear antigen showing co-labeling with SARS-CoV-2, documenting this is, obviously what we saw in ISH kind of confirmed that they were present in neurons, but this makes the final confirmation. And then this image is, the top image is uninfected mouse, just showing the normal expression of GFAP and IBA1. So IBA1 for microglia, GFAP for astrocytes, and then the bottom image showing that there's increased both of them. I mean, obviously the IBA1, we saw that on the H&E, that there was increased microglia, but we didn't necessarily appreciate increase in astrocytes in the infected animals, so this kind of shows that. And so there's a lot of groups out there working with this model now, and when we published this article, there's the article I mentioned earlier. There's a bunch of other groups that published articles about the same time or since then, and this here is one of them. This is was slightly different than ours in that they used a higher dose and all the animals succumbed, whereas we had out of 28 animals, we had 4 survivors. And so there's kind of a postulation that the virus dose probably does influence outcome. And one other difference here, they saw virus in the choroid plexus. They have some nice images in this article showing virus in the choroid plexus by IFA. And we didn't see that. And after I read this, I went back and looked at all our slides. Like, oh my God, did I miss something in the choroid plexus? And I didn't see anything in the choroid plexus in our study. But they found it here, and then their findings were very similar to ours. And then they also said vasculitis as well. And then this is another article that came out recently. This is actually from the group that originally developed the model, which I'll go into in just a second. But they had 3 different dose groups. All their high dose animals succumbed. All the low dose animals lived, but they didn't get universal infection in the brain in this study. But this is a neat study because they have a very detailed chart at the end of this article detailing kind of where the virus is, kind of sub details about where the virus is located in the various regions throughout the brain. So this is a really good article. But one other thing, they have a little bit more detailed analysis of the olfactory epithelium. And obviously it's very complex. There's the neuronal cells, the sensory cells that send their axons through the cribriform plate into the olfactory bulb. There's the sustentacular support cells. There's stem cells there, and there are Bowman's gland cells in the olfactory epithelium. And they located the virus to the sustentacular cells as opposed to the neuronal cells. And then they had a detailed discussion of the anosmia and part of the study was dedicated to that. And so, as I mentioned earlier, this model was developed by Dr. Perlman and as a model to study the original SARS-CoV back in 2008. And so this was interesting. Again, they have a detailed chart in this, a detailed table in this article that identifies where the virus is located throughout the brain in a lot of granular detail. But they didn't see any inflammation with the original SARS-CoV in this model, but they did see a lot of virus in the brain, and they did see neuronal loss. And this article preceded the last one by the same group. Again, this model was developed at University of Iowa by Dr. Perlman. Dr. Marholtz was the pathologist on this original study. And this article details the development of this mouse model, and the presence of the K18 promoter is necessary for epithelial cell specificity. And to the question earlier, the ACE2 expression was documented in these organs in a much lower level in the brain. And in fact, in the third, they had 3 different lines with different levels of ACE2 expression, and it was least in the 3rd line. And even though the human ACE2 receptor was expressed at very low levels in the brain in the 3rd line, there was still abundant virus present. So the level of virus is definitely not commensurate with the level of human ACE2 receptor expression in the brain. And so it seems that there are other ways for the virus to get into the brain and spread. And that's kind of a nice segue to this next article. So kind of in order making the case for why this model is relevant to how the disease occurs in humans, I did a review of the literature, and this is an interesting article that talks about viruses that can use the olfactory route to infect the CNS. And so these are all non-coronaviruses, but everything from influenza to West Nile virus potentially, and it proposes routes of infection via the olfactory respiratory neurons in the olfactory mucosa that send their axons through the cribriform plate. And this is a great article, but because it proposes a few different routes of infection via the olfactory route of infection, not only via axonal transport, but also via these olfactory ensheathing cells, which is basically the cells that sit in the cribriform plate and kind of guide the axons through the cribriform plate. And then once it's in the CNS, the article also proposes routes of spread throughout the CNS by these viruses. So this is definitely a route of infection that is been talked about. It's just the details of how it occurs are not well known, but it seems to not necessarily be dependent on the presence of the actual virus receptor. And so this, again, making the case for why this model is relevant, these are some of the findings that have been described in humans with COVID-19, albeit not necessarily common compared to some of the respiratory features, but definitely have been described. And I'll just draw your attention to this second bullet point. Inflammatory CNS syndromes, they described in this article, high incidence of acute disseminated encephalomyelitis with hemorrhagic change, unrelated to the severity of respiratory disease. And then the third bullet, ischemic strokes. I read this, when I read through this, I was like, man, it's kind of similar. I could see some of these findings fitting with what we're seeing in this model in the brain. Again, more on what they're seeing in neurological associations of COVID-19. I put this article in because of the first bullet here, where they describe the CNS, that they, other coronaviruses they found in the CNS, including SARS-CoV and MERS, but also human seasonal coronavirus. So I believe there's 3 human seasonal coronaviruses that just cause mild cold-like symptoms. And there's a Journal of Virology article from 2000 that describes the presence of 1 or 2 of these seasonal coronaviruses in the brain. And they were looking to draw an association with multiple sclerosis. But when I read that, I was like, man, that's really interesting that the seasonal coronavirus would also be in the brain. So just really, I think makes this model very relevant. And then finally, I mean, there's so much in the literature that comes out on this all the time. It's hard to keep up, but this article was one I pulled because it actually had autopsy cases of patients that died of COVID-19. And these patients didn't necessarily have neurologic syndromes or symptoms or findings, but they did have COVID-19. They had 33 individuals in this study. And they found, as I mentioned earlier, the one study with this mouse model found the virus, they located it to the sustentacular cells in the olfactory epithelium, whereas this study actually double labeled the olfactory epithelial and neural cells. So essentially the sensory cells in the olfactory epithelium that send their axons into the olfactory bulb, they actually co-located the virus to those cells as opposed to the support cells. And then also one other point from this article, they actually located documented the highest level of viral RNA directly below the cribriform plate. And I mentioned those cells that kind of sit around the cribriform plate and kind of guide the axons through. So I just think this is a great article. Again, one of the first ones I saw of nice CNS lesions in actual autopsy individuals who died of COVID-19. And so that concludes. So I just wanted to show this slide. The next few slides are similar to what I described in the main slide. But there was only one day 11 animal in this study. Most of the animals succumbed between days 5 and 7, and this was the day 11 animal, and that had the worst degree of kind of hemorrhagic change in the CNS. So I wanted to show an image of that. And very similar to what I described with the apoptotic or necrotic neurons. And I don't know how we're doing on time. I have
just finish. Okay, you can finish with this one. I'd be interested to see what people say.
Okay, so I put these slides in here to kind of get other individuals' thoughts on it. So during my description, I kind of went to this area of the cerebral cortex and I described these neurons that were vacuolated, and I interpreted that as a degenerative change. I'm interested to hear what others think of that. So there are these neurons, and they're, I would say, multifocal minimal distribution. So there's not that many of them, but they have this abundant, clear, almost kind of granularish foamy cytoplasm with a single large vacuole that often compresses the nucleus. So I know vacuolation is certainly described as a degenerative change in virus and in the CNS, but there's at least one apoptotic or necrotic neuron here. But I was kind of unfamiliar with this change, and so I interpret it as degeneration, and I reached out to some colleagues who thought it might also be a degenerative change. But we also did some EM, we got some EM images, and this is what, a lot of this is kind of swollen endoplasmic reticulum. But these large clear vacuoles, this was taken from a box, so it's unclear to me, I mean, this change was seen on the CNS, it's unclear to me if this represents a real change or an artifact. We were just not really sure. So I'm kind of interested to get other folks' thoughts on what they think of these changes. Again, let me show, yeah. So the swollen ER does have particles. I mean, the size of the virus particles of SARS-CoV-2, I think it's documented 40 or 60 to 140 nanometers. And some of these particles that we saw in this swollen ER are approximately the right size.
You didn't see any RNA or antigen in those cells, did you?
Well, it was really hard on the RNA scope, but there was some detail that was lost, on the cellular detail that was lost. So it was really hard for me to definitively co-locate virus in those vacuolated cells with the RNA scope images. But there was
IHC could work. There's good IHC for.
Yeah, we may look at IHC. We haven't done that, but that's an excellent point. But there was abundant virus in the area. I mean, it's very similar to the ISH image I showed. There was just abundant virus in this whole area, but as far as seeing the detail of these vacuolated cells, I just couldn't see it. And we may do some gold labeling on these EM images, but that's kind of what we're seeing. I was just interested to get other thoughts on what they think of this change and whether they think it's non-specific degenerative change or perhaps even artifact.
Okay, so just go
Almost
back.
certainly got to be related to viral pathic effect, but it'd also be interesting to know if those are monocyte-derived macrophage type cells, or if those are actually infected and degenerating neuron cells.
Well, no, they look like neurons, I think. Go back to the H&E and take a look again. Anyone? Okay.
You may be right, Jerry, but I'm always fooled by how
Right.
protean macrophages are.
Okay. So it's getting late. We'll have just a comment or two on those cells.
Well, this is as everybody pointed out, and Curtis, I actually recommend you talk to Raul. He had some good points in the chat box, and I think there's, if I get my thought processes in order, I think there's some studies to be done, and maybe you want to use this mouse, but you also want to correspond. We heard about the mouse adapted CoV2, which doesn't require a transgene for the human ACE. And I think that ultimately findings will be a little bit different, but maybe there will be some important similarities, especially in the nervous system, which I'd like to know more about. And then, what happens in the setting of vaccination, because this is an unusual population of cells, right, to infect as a virus of essentially non-dividing cells. And so in the context of vaccination, how well do you prevent this transmission to neurons?
Right.
So you're not getting the vaccine then, right?
No, I'm getting it.
Okay, well, I think that's about it for today. We got so many things to talk about, a lot of good questions there. And so I'll hand it over to Sandy. Thanks to all the speakers. They're three great cases,
Yeah, great cases. Thanks for everybody for staying a little bit late. This will be posted as a video on the website. I'll send by email the website link to remind everybody, but it's centerforgenomicpathology.com, and then sublinks within there will get you to the videos. And we'll see you in January. Be healthy and happy.
Stay safe.
All right, great.
Bye.
Thank you. Thank you all for having me and letting me do the case. Appreciate it.
That's great.
Thank you.